Today, we are diving deep into one of neuroscience's greatest mysteries, schizophrenia. And what's so fascinating is that the clues, well, they start appearing long before a person is even born. It's really a kind of prenatal detective story.
So think about this for a second. This is the central question for our whole investigation today. How in the world do you solve a mystery when the key events happen during pregnancy, sometimes decades before any symptoms show up? Our story begins with a series of really puzzling clues. Clues that weren't gathered from a crime scene, but from huge studies of people all over the globe. For years, scientists have seen these strange connections. Kids whose mothers had the flu or went through a famine or experienced some kind of intense stress while pregnant. They have a statistically higher chance of developing schizophrenia. But here's the thing. Those are just correlations. They're vital clues, for sure, but they don't tell us how or why. It's not proof. And that leaves us with this massive question. To really connect those dots, to go from just a correlation to actual causation, scientists needed to find a way to peek inside the ultimate black box, the developing brain of a fetus. But how could they ever do that? Well, this is where our scientific detectives come in. The key to cracking this whole prenatal case, it lies in something called preclinical animal models, and they are absolutely central to the research we're talking about. So what exactly is an animal model? Think of it like a stand-in, usually a mouse or a rat, that researchers use to understand a human disease. And here's why it's so crucial. These models let scientists run controlled experiments that would be completely unethical, just impossible to do with pregnant women and their babies. And a process they follow is incredibly logical. First, you start with that clue you found in the human studies. Then you carefully recreate that situation, like a simulated infection or a very specific diet in pregnant animals. After that, you study their babies as they grow up, looking for any changes in their brains or their behavior that look similar to the human disorder. And then, the final step, the grand prize, you try to pinpoint the exact biological mechanism that connects that thing that happened during pregnancy to the changes you see later in life. Okay, so with that in mind, let's open up our first case file, the infection. We're looking at the link between a mom getting sick during pregnancy and the risk of schizophrenia in her child. So the clue from the real world, from all that human data, is pretty clear. But what's really going on here? What's the mechanism? Okay, so in the lab, scientists can trigger a mother's immune system without actually using a live virus. They call it maternal immune activation, and the findings are frankly remarkable. The offspring show abnormal behaviors. They have changes in their brain structure and chemistry, and get this, they even respond positively to the same antipsychotic drugs used to treat people. This is just a brilliant example of how the animal model can confirm and then build upon that initial clue from humans. And this work led to a huge breakthrough. It turns out it's not the virus or the bacteria itself that's causing the problem. Nope. It's the mother's own body, her immune response to that infection. That is the smoking gun in this case. All right, let's move on. Time to open our second case file. This time, our detectives are looking into the role of prenatal nutrition. Now, the clue here comes from these really tragic, large-scale events, like the Dutch hunger winter during World War II, and also from studies on specific vitamin and mineral deficiencies. And the data is consistent. Kids whose mothers went through these hardships have a documented higher risk. So back to the lab, scientists recreated these exact conditions, putting pregnant rats on diets that were lacking in either protein or vitamin D. And once you know it, the pattern was starting to look really familiar. The offspring had altered brains, and as adults, they showed major behavioral changes that look a lot like symptoms of schizophrenia. It's another powerful piece of evidence pointing in the same direction. Okay, for our final case file, we're going to examine a clue that's maybe a little harder to pin down: the impact of severe maternal stress on the developing fetus. Again, the link in humans is there. We can see that major stress during pregnancy is associated with a higher risk. But how on earth could a psychological state, a feeling, biologically impact a developing baby? The animal models basically blew this wide open. When pregnant rodents are exposed to stressors, their offspring are born with a whole cascade of biological changes. Their own internal stress system, the HPA axis, is basically thrown out of whack. Their immune system is altered, and you see physical changes in parts of the brain that handle emotion and learning. This isn't just in their heads, it's a deep biological connection. So we've just looked at 3 totally different case files, right? We've got infection, we have lack of nutrients, and we have stress. On the surface, they seem like completely separate things. But are they really? I mean, what if they aren't separate at all? Could there be a common thread, a single biological culprit that's pulling the strings and connecting all these different prenatal events? And here it is. This is the big twist in our detective story, the one key insight that ties all the evidence together. It looks like a huge amount of research is pointing to a single common pathway, maternal immune activation, the very same thing we saw in our first case file. So how does this all connect? It's like a biological chain reaction. We know an infection kicks the mother's immune system into gear. But what's really wild is that animal models show severe stress can do the exact same thing. And when the immune system gets activated, the body releases these little messenger proteins called cytokines. Here's the kicker. Those cytokines can cross the placenta and directly change how the fetal brain is being built. This could be the unifying link between all these seemingly unrelated risks. So why does cracking this case matter so much? Because it completely changes our focus. It helps us move from just trying to treat a disorder after it's fully developed to maybe, just maybe, preventing it from ever happening in the first place. Understanding these super early origins could open up a whole new world of strategies that are all about ensuring a healthy prenatal environment. So as we wrap up, I'll leave you with this idea to think about. For decades, the entire approach has been about managing symptoms after they appear. But all of this research forces us to ask a really profound new question. Could the ultimate key to this complex disorder lie not at the finish line, but way, way back at the very beginning, in those 9 months that literally shape a lifetime?